Good afternoon, everyone, and welcome to our webinar on VHH discovery by Phage display. Our speaker this afternoon is Dr. Sam Sugerman, who is the principal scientist at Abcore, leading our VHH discovery services. And today he's going to be talking to you about VHH and the discovery pipeline that we use at Abcore. Thanks, Sam. I'll leave it over to you.
Thanks, Aaliyah, and great to see everyone virtually. Today I'm talking to you about VHH discovery using our Abcore product line. Here at Abcore, we are a deeply experienced team. We've started around 2011 doing mostly polyclonal antibody discovery in smaller animals. And since about 2015, we moved into an animal-based immunization platform using llamas and alpacas to discover single-domain antibodies known as VHHs. Our brands under Fortis Life Sciences include the Antibody Solutions Unit, my team Abcore, as well as Arista Biologicals and Bethel Life Sciences. We also have Empirical Bioscience and our diagnostic divisions of IPOC, and then Nanocomposites, which is a nanomaterials distribution and manufacturing team, and Vector Biolabs is our cell and gene therapy space. Here at Abcore, we do an immunized discovery for VHH antibody development, primarily for therapeutic, but we've also done some diagnostic, some bioprocess workflows as well. And here our team uses primarily a phage display workflow, though we are branching out into alternative custom vectors as well. Starting briefly with a high-level understanding of the VHH, in camelids, the antibody response has evolved to have two main isotypes. The first is sort of your canonical IgG, where you have a paired heavy chain and light chain at the interface of the variable region to form your paratope. Many years ago, camelids evolved a second isotype called a heavy chain only antibody, where essentially the CH1 region that is responsible for the pairing of the light chain was removed and truncated and replaced by an elongated hinge region. And what this has allowed to take place is a evolution over many years of what we call the variable heavy chain from HCABs or VHH domains. And so this is a variable domain that exists in an unpaired interface. And what that allows it to do is exist sort of as a standalone subunit where it will bind without additional context from a paired interface. It's generally more amenable to truncation and daisy chaining on some level. And it also exists in a more soluble interfacial framework. One of the sort of real assets of these VHHs as it relates to therapeutic discovery is that the length sampling in CDR3 is quite similar to that of humans, also quite similar to that of rabbits for those who've done polyclonal IgG development. And then it's very valuable as a protein engineering scaffold for its engineering potential into multiple frameworks, such as a bispecific antibody, for example. When we're looking at the comparison between VHH and the combination VHVL, we are very interested in what the sort of artifacts of being an unpaired VHH interface will do to the sequence of that variable domain. And this is something that's been explored in literature. One of the great structural papers that has compared this came out of this 2018 paper with the data pictured. And really, to me, the really interesting thing here is that you have a pretty significant difference in the RMSD, sort of the conformational flexibility on some level between the antigen bound in CDR1 as well as the antigen bound in CDR3. And you can see a very significant difference between the antigen bound VHHs and the antigen bound VHs in orange. And so, one of the real high-level takeaways here is that we have more variability in conformation in an antigen-bound VHH than you might see in a comparable VH framework. One of the limits, however, because you only have the half of the interface from the VHH and you don't have the paired VL, is that there is a much lower surface area, especially a solvent accessible surface area measurement. And so this really limits the ability of the scaffold to form things like pockets for small molecule binding or disordered region stabilization. And it also really limits the ability to specifically recognize hydrophobic surfaces, because there's just sort of a limited solvent exclusion potential. And so you're creating sort of a greasier interface on some level if you're going for something very sticky. Here at Abcore, we have recently moved into a discovery library space, and one of the things that we really like to clear up early on is the difference between our discovery library services, such as this AbNano VHH naive library, and our immune discovery workflows. And so one of the biggest things that we have noticed a difference on is the affinity of a typical hit coming out. And so how these two different pipelines work on a very high level, the naive library is a pre-existing cloned library taken from non-immunized llamas and alpacas in our herd. The custom VHH discovery pipeline services will immunize animals and allow the animal to mature toward a high immune response and binding of those specific antigens. And so what you see is the results of that maturation process on some level, where you have on the order of about a 50-fold difference in affinity between the discovery libraries and the custom discovery services. The sort of high-level applications of this are actually very different, where they're unique strengths. So one of the things that has recently come to light in the immune therapeutic space is that lower affinity binders may actually be necessary, because on some level there's a fast cycling time and lower affinity necessary to create these really efficient bispecific T-cell engagers, for example. So you don't want high affinity to something like CD3. And so to have a sort of weaker and more transient interaction is actually advantageous here. For applications such as payload targeting, or if you're really trying to get a really high time fraction inhibition of a cryptic epitope, you're going to want something very, very specific. And for that, we would really recommend going with the custom discovery services. The other sort of advantage of the discovery libraries is because they're not biased by the immune system toward a particular antigen, you can use them to target many different antigens. Whereas if you are immunizing an animal and taking it through a full campaign, you're only going to have a good binding response to your immunogen and things that have very high homology to your immunogen. For our number of animals used and essentially the diversity of the resultant library, we're going to be able to use a lot of animals to generate our discovery libraries, and that will create a really nice profile for the discovery services. But for the custom immune workflows, we're only able to usually do a couple animals, because it's cost prohibitive on some level to do larger things. If there are very desired large animal projects, we can certainly take those on a case-by-case basis. And then really one of the huge selling points and takeaways here is that the timeline for panning a discovery service is very quick. And so the panning fraction of our campaigns is only about a month. If we're starting with a pre-cloned discovery library, then we're able to accelerate the project significantly. If we have to wait for the lag phase of immunization, then it adds a significant amount of time. So we like to say that these discovery libraries that are pre-existing and pre-cloned are about 18 weeks faster. We're going to talk very briefly about our immune discovery services. Looking at our pipeline at a high level, we initially have an immunization with our antigens, and our immunization campaigns typically have on the order of 5 immunizations over the course of about 15 weeks. We're isolating the pooled mononuclear lymphocytes from there and taking all of those as a pool through to constructing a phage display library. This phage library is taken through preliminary validation, and then from there, panning enrichment against the antigen of interest over the course of multiple rounds. We take that to a single point ELISA using an off phage system, so we're able to sort of evade all of the artifacts of displaying VHH on the phage coat protein. And so we're able to screen a free but crude VHH as an initial screen. And from there, we can do downstream assessments like expression purification, cell binding, mammalian expression, etc. Common immunogens that we will handle on our team and sort of to help shape the scope of work. We really prefer recombinant proteins. And one of the interesting things that we've noticed with VHHs as a scaffold is that they perform much better in recognition of a target epitope that has sort of a ternary nature, where you have some sort of high order and structure. Maybe it's something like a cleft or a binding pocket. Maybe it's something as simple as a helix loop, helix motif. But to sort of play to the strengths of the scaffold, remembering that we can't create pockets and we can't sort of deeply interact with smaller disordered areas of epitopes. We really want a nice ordered or rigid surface that the VHH can interact with. We do have quite a bit of experience with VLPs, liposomes, as well as nanodiscs. And then we've also immunized with cells and sort of created those complex environments as well as sort of an immune stimulation. And then more recently, we've had some good successes with DNA, both with and without electroporation, to deliver it into the animals. One of the things that we really like to consider from immunization is how the antigen design will create a successful project. And so things like homology to other FCs, homology to any hinge regions, and then homology to any albumins, as well as sort of pre-existing immunodominant motifs. We've noticed GST, for example, is very immunodominant. We can create cross-reactive binding profiles. We can create selective binding profiles. All sort of from immunization all the way through, and we have quite a bit of experience doing that. As I said, our typical immunization timeline is about 5 injections over the course of about 15 weeks. From there, we're isolating typically 2 production bleeds with PBMCs. The rough yield of PBMCs is on the order of about 5 times 10 to the 8th. And from there, the fraction of VHH producing B cells is on the order of about 20%. And so for 2 animals, 2 bleeds each, we're looking at a library size for theoretical diversity somewhere on the order of 5 times 10 to the 8 immune matured VHH molecules. Looking a bit more closely at our library construction procedure, we're going from our PBMC pool. We go through a total RNA workflow. There's an RT-PCR step, and we're able to isolate very specifically the VHH band of interest. From there, we're taking our VHH into a commercially available phagemid vector. We're using a PADL phagemid, and we're transforming into TG1s before our preliminary library QC step. From there, we take our libraries through phage panning to enrich the binding pool. Abcore really prides itself on having a number of different phage panning resources under one roof. We have lots of experience in capturing different binding profiles, as well as unique questions such as selectivity or cross-reactivity or live cell binding. Here you can see an example of a panning campaign where we were able to enrich on the order of about 3.5, 4 orders of magnitude after round 3 panning for our antigen of interest relative to an off-target control. And finally, our typical final step in the campaign is an off-phage initial screen, where we're doing a single point ELISA against the antigen of interest, as well as any cross-reactivity monitoring or homolog binding monitoring to similar proteins of interest. We really pride ourselves on taking this off-phage so that we get a cleaner answer to binding questions. This is a crude periplasmic shockgate, and we're able to screen against either recombinant antigen that is fixed through a couple different mechanisms, or using a live cell by flow cytometry. From there, any sequences are measured, and the typical deliverable for this pipeline is sequences to the customer. For an example of one of our immune projects that we have done internally, we immunized with an anti-human FC. And from there, we were able to see a very high heavy chain only antibody response here on the serum ELISA. You can see that even after the first bleed at 6 weeks, so after 3 immunizations, we have very strong serology. From there, we took it through panning using a biotinylated FC and a solution phase panning workflow. This is our single point screen, including our positive control. All the way through, we isolated 80 unique hits from the 96 clones that we screened. And from that, we were able to run some purified EC50s of the final VHH candidates. And we're getting sub-nanomolar interactions measured by ELISA. Through a separate campaign, we did a human CD4 ECD. Again, you can see pretty early response for the heavy chain only antibody conjugates from bleed 1 having a reasonably high serology, though admittedly not quite as high as the IgG FC. You can make some arguments about immunodominance or sort of immune response for other species FC in this case. And from there, we were able to take some really high affinity measurements at the library stage as well as a validation for the library cloning step. One of the binders that we isolated to date on this is as well sub-nanomolar. From here, I'd like to spend just a few minutes talking about our recent product launch, the AbNano VHH naive library. This library was taken as a way to accelerate our workflows. We wanted to really offer an option that let us fast track discovery. As well, we wanted something that was a much broader scope in application, where it wasn't sort of matured against one or a few different antigens of interest. And so to do that, we took 103 naive camelids from our herd, and we isolated production bleeds from those animals. We ended up with about 1.5 times 10 to the 10 ppmcs. We built this out as 13 sub-libraries, and the reason for that was mostly from a workflow perspective, but it really had the advantage of allowing us to avoid artifacts of construction. A pooled sub-library approach is going to limit sort of PCR bias, et cetera, and similar sort of redundancies from the construction artifacts. From there, we put it through our in-frame and library QC. We ended up with something on the order of 89% in-frame VHH by Sanger. And we also sent for long-read NGS for each of the sub-libraries and pooled those metrics. Here we're presenting just sort of a brief snapshot of the NGS metrics from these sub-libraries. We're seeing that there's a nice CDR3 length distribution. This is a bit left-leaning relative to some CDR3 length distributions that exist in literature, and our rationale for this is that this is a naive portfolio rather than an immunized animal pool. And so our rationale is that there's a little bit of right shift on this curve in the presence of some of our immunizations. And we're currently working up some data that demonstrates that empirically. There's also a very limited redundancy between the sample sets of naive animals. One of the concerns as we started building this as sub-libraries was that there would be a lot of overlap between the different pools of animals on some level. And so what we were able to measure was the cluster diversity of the sequence on an amino acid level. And so here on the right, you can see that we have very low frequency of clusters. You see just a little blip there at about 10 for the cluster size. But what that's demonstrating is that there's not a lot of redundancy from one animal to the next, and that fits well with what's described in literature. The other concern that we had, especially as we were doing this using a MySeq NGS technology, is that some of our diversity metrics would be artificially inflated by something like based call, for example. And so we ran a bandpass filter with a nucleotide analysis using a 95% homology filter. And you can see that there's really good agreement actually between the 95% homology on a nucleotide basis and the amino acid unique basis. And so we're really happy with these measurements, and based on sort of the shape and distribution of this number, we are pretty confident that we are not fully sampling our library with this sequencing read. One of the ways that we really wanted to validate this naive library was through a few different panning campaigns. And so we started by developing different data sets against a few molecules of interest that were interesting to us and to others. To start, we performed some panning campaigns against a couple extracellular domains of RTKs. We targeted the insulin receptor with the beta isoform, and then we also targeted an EGFR extracellular domain. And finally, we chose to really focus on human PDL1 for the immuno-oncology applications as well. Our panning campaign against the insulin receptor ECD yielded on the order of 47 hits that were measured unique on an amino acid basis. This was performed using a solid phase adsorption, and so there was a lower concentration of target antigen at each round, as well as a sort of random orientation of target antigen at each round. Between each round, we were using Phage precipitation to overrepresent any rare clones, and from there we characterized a couple very common binding motifs from the sequencing, and those were within a couple orders of magnitude. The lower end was on the order of 30 nanomolar. The higher end was on the order of about 500 nanomolar, both measured by ELISA. From there, we did a panning campaign against EGFR, the extracellular domain. One of the things that we really wanted to target with this extracellular domain was we wanted to see how quickly we could get through panning and into screening. And so from the naive library, we panned using supernatant at each step with no amplification between rounds to improve the speed of the workflow. We were able to pan Monday, Tuesday, and Wednesday, and we were able to mini prep and transform on Thursday to start screening on Friday. All within 1 week, all 1 FTE all the way through. From there, we did notice that there was a lower unique hit rate with about a 7% unique hit rate on an amino acid level. And we were able to express, purify, and characterize the most abundant clone, which ended up having an EC50 on the order of about 200 nanomolar by ELISA. And so our takeaways here are, that this is a great workflow if you need something to bind very quickly, and if you are time limited in your pursuits, but it does seem to impact the unique hit rate. Finally, we really wanted to flex the strengths of the library and see what we could get out of something that is a high value target like PDL1. And our goal for this panning campaign was to really capture all of the rare clones and see how diverse a profile we could pull through side by side in one workflow. And so what we ended up doing was 3 rounds of solution phase panning using Phage precipitation of each panning output between. Elution was by triethylamine. And then we ended up screening 5 96 well plates of clones. We captured 147 hits. Of those, 68 were unique, which represents about a 14.5% unique hit rate. We took a random selection of the different single point ELISA signals, and from there we were able to isolate what ended up being 3 orders of magnitude difference, EC50, and so we were able to capture single-digit nanomolar and single-digit micromolar hits using the same workflows all side by side, which was really impressive to us. And so to sort of look at the high-level conclusions, our immunized workflows are able to generate single-digit nanomolar binders or even sub-nanomolar binders against specific antigen of interest or family of antigens. The IP for all of these custom discovery workflows is novel, and Fortis doesn't withhold any assets for the custom projects. There's something on the order of a 5 to 6 month workflow between the immunogen delivery to our team and the sequence delivery to your team. For the discovery libraries, the first to market is our large naive discovery library described, and we're getting somewhere on the order of 50 nanomolar binder as sort of the median for our isolation. From there, we are able to offer unlimited licenses for individual sequences or sequence families as part of the intellectual property. And we're currently set to offer a 6-week turnaround time from antigen delivery to sequence delivery using the discovery library workflows. And And with that, I'm happy to conclude this presentation. Thanks all for your attention.
Thanks so much, Sam, and thanks to you all for listening. If you're interested in learning any more about the library, the VHH services from Abcore, or anything else Sam discussed in his presentation, you can find the links in the description for this webinar. And with that, thanks everyone and have a great afternoon.